the mapping population using SSR marker. Genotyping is the identification of markers associated with our desired trait. Now let us see the steps involved in genotyping. The first one is selection of mapping population, second one DNA isolation, third one PCR analysis, fourth one page, and fifth one is scoring. First, let us see about the mapping population. The mapping population that I have chosen is the F2 population of maize. The trait is downy mildew resistance. The gene responsible is present in chromosome number 2. The locus is 2.04. This is the diagram showing chromosome number 2 of maize. The gene of our interest is present in the fourth pin of this chromosome. The parents are female UMI 79 and male UMI 936. This is the female population which is downy mildew susceptible. And this is the male population which is downy mildew resistant. And this is the F2 population that we are going to genotype. The next step is the genomic DNA isolation. These are the overall steps involved in genomic DNA isolation. Collect about 1 gram of leaf sample from the F2 population and also from the parents. Cut it into pieces. Add 400 microliters of CTAP buffer and grind the leaf tissue. Now centrifuge the mixture at 12,000 rpm for 20 minutes at 4 degrees Celsius. Now take the supernatant and discard the pellet. Now add equal volume of chloroform isoamyl alcohol to the supernatant. Centrifuge it at 12,000 rpm for 20 minutes at 4 degrees Celsius. Now we will get 3 layers. The topmost aqueous phase contains the DNA. To the aqueous phase add equal volume of ice cold isopropanol and again centrifuge it at 10,000 rpm for 20 minutes. To the pellet add 70% ethanol. Again centrifuge it, add dry the pellet and dissolve it in 50 microliters of DE buffer. For checking the quality and quantity of the DNA that we have isolated, agarose gel electrophoresis is done. About 0.8% of agarose gel was prepared and the DNA samples were loaded along with the dye and the gel was run. The image can be viewed under Alpha Imager. The next step is the PCR analysis using SSR marker. This is the materials required for the PCR. This is the PCR cocktail for 1x. The total reaction volume is 15 microliters. First, the PCR buffer is added. The PCR buffer contains magnesium chloride, tris HCl, KCl, and gelatin. After adding PCR buffer, the DNTPs are added. The primers, both forward primer and reverse primer of 1 microliter is added. Next, the genomic DNA is added. At last, the Taq polymerase is added. The PCR tubes are placed inside the PCR machine. This is the PCR conditions for maize. These PCR conditions are now set up and the PCR machine is run. The next step is the urea page. First, clean the glass plates with 70% ethanol. Coat the IPC chamber with rain repellent and coat the outer glass plate with bind saline. Wipe the side clamps, spacers, combs with 70% ethanol. Place the spacers on the IPC chamber, then place the glass plate on it. Assemble the unit with side clamps, bottom assembly, and lock the unit. Insert the comb between the plates. Now 10 ml of gel matrix is prepared, which contains 40% acrylamide solution, 10 ml TE buffer, 10% APS and timid. Inject the solution and make it spread uniformly and immediately insert the combs. Now allow it to polymerize for 1 hour. After polymerization, keep the assembly in a base chamber filled with 1x TBE buffer. Remove the combs, then pour the TBE buffer between the plates and the IPC chamber. For 20 microliter of PCR product, 5 microliter of page dye was added. The samples are loaded in the wells. Now the gel is run at 100 watt for 1.5 hours. The dye contains bromophenol blue and silane cyanide. They run at the same speed as the DNA fragments, which are less than 5 base pair in length. The movement of dye along with DNA can be visualized here. After 1.5 hours, disassemble the unit. Gel plates are subjected to silver staining. The gel plate was first stained in fixer for 15 minutes. The fixer contains 200 ml of acetic acid and water. Then washed with double distilled water for 5 minutes. Then it is kept in staining solution. The staining solution contains silver nitrate 2 grams and 3 ml of formaldehyde and water. Again the gel plate was washed with double distilled water. The gel plate was again kept in fixer and again washed with double distilled water. The gel is then air dried and documented. This is the urea page gel of our F2 population. The next step is scoring. The urea page gel is scored depending upon the polymorphic bands shown by the F2 population in relation to its parents. An Excel sheet is prepared with the polymorphic bands shown by the F2 population in relation to its parents. By scoring, the downy mildew resistant gene frequency of parent in the F2 population is studied. That is, the average proportion of parental genome in F2 population can be studied. Through the MapMaker software, the scored data can be used for linkage map construction. This is the linkage map constructed for downy mildew resistant in maize.